all, this video is about gene regulation in eukaryotes. As we talked about, eukaryotic cells like humans or plants or fungi have many different cells, and they need their cells to do different jobs. So the way that we make sure that cells can be specialized is each cell reads a different part of the DNA and uses that part of the DNA to make different proteins. And that allows cells to become more specialized. How does that actually happen? Let's talk about some gene regulation strategies in eukaryotes. First of all, I want to talk about gene regulation strategies that occur in the nucleus of the cell. The first example we'll look at is DNA chromatin. Chromatin is the uncoiled form of DNA, but when I say uncoiled, I don't mean completely uncoiled. Chromatin is still DNA coiled around histone proteins. So, inside a chromosome, you have tightly coiled DNA. Then you can also have chromatin. In this instance, here's the DNA. I'm labeling that in pink. Okay. Inside of the DNA is this histone protein. That's helping to keep the DNA organized and to make sure the DNA doesn't tangle. When the DNA is tightly coiled around the histone protein like this, okay, there's no transcription that can occur. So when the DNA is tightly coiled, we can have no transcription because the RNA polymerase has nothing to attach to. So what happens if we want to see this gene to transcribe and translate it? We can do something called histone acetylation. In histone acetylation, this turns on our DNA. And what happens is we add an acetyl group, COCH3, directly to the histone protein. That allows the histone proteins to separate from each other so that the DNA becomes less coiled up and you can see the genes here. So here again is the histone protein. When the DNA is tightly coiled around the histone protein, there's no transcription. When the DNA is less coiled around the histone protein, there is transcription. So that's why we say the DNA is turned on, and this means transcription can occur. So these acetyl groups basically move the histone proteins apart so RNA polymerase can attach and transcription can occur. So again, acetylation turns genes on. What about if you want to turn genes off? You have to do something called DNA methylation. DNA methylation turns off the DNA. How does this occur? Well, again, if this is your DNA right here, your DNA is made up of a bunch of nucleotides, A, T, G, and C. In DNA methylation, we add methyl groups, CH3. These methyl groups attach to the outside of the cytosine nucleotides, and this prevents transcription because RNA polymerase can't bind. So how do acetylation and methylation affect protein synthesis? Well, they affect whether or not the first step in protein synthesis can occur. They affect whether or not transcription can occur. Acetylation allows transcription to occur. Methylation blocks transcription. And by activating or deactivating different parts of the DNA, we can control what proteins are made. There's another example of something that helps increase or decrease transcription, and we've already talked a little bit about this. These are the transcription factors. This is also something that occurs in the nucleus of the cell. Transcription factors are proteins, and there are two main categories of transcription factors. Remember, transcription factors are proteins that bind to the promoter sequence on the DNA, allowing the RNA polymerase to bind. There are general transcription factors that allow some transcription to occur, and then there are more specific transcription factors. Okay, So let's start by talking about the DNA. Again, if this is the DNA right here, in pink, this is the gene right here. This is the thing that codes for the protein. The promoter region doesn't code for the protein, but it's where the transcription factor is attached to. Okay. If you're before the gene, this is known as the upstream area. If you're after the gene, you're downstream. Okay. So general transcription factors are going to bind near the promoter sequence so RNA polymerase can bind. There are other transcription factors called activators. Activators can upregulate, meaning they can increase transcription. And activators are a specific type of transcription factor that attaches upstream on the DNA. That means before the gene we want to transcribe. So if you have transcription factors that are activators that attach upstream of the promoter sequence, you have general transcription factors that attach to the promoter, you're more likely to get RNA polymerase to bind, so you're upregulating, you're increasing transcription. There are also silencers. Okay? Silencers or repressors. These can also bind upstream on the DNA, and these are going to downregulate or decrease transcription. Okay. Both activators and silencers are chemicals. They can be proteins, they can be other things. These are chemicals. And they can be hormones or other types of proteins. Okay. Basically, what these activators and silencers do is they influence how much RNA is transcribed for a certain gene. So for example, if you want to make more hemoglobin protein, you would make more hemoglobin mRNA. Right? So those are transcription factors. Again, that occurs in the nucleus. The last nuclear example we'll talk about is RNA processing. We've already talked about RNA processing, but basically, okay, the 5' cap, the poly A tail, the introns and exons being removed, that can influence the mature mRNA. Okay? So one thing that can influence mature mRNA, as we know, is this idea of alternative splicing. And this allows you to create many different mature mRNAs from the same DNA. So that's one way RNA splicing can regulate genes. Another thing that can happen is the length of the poly A tail. Longer poly A tail means longer life. So more translation can occur. So the length of the poly A tail actually influences or determines the lifespan of the RNA, because the poly A tail is protected. It doesn't code for anything, but enzymes, as soon as this RNA leaves the nucleus and goes to the cytoplasm, enzymes are going to start destroying the RNA. So if you have a longer poly A tail, this mature mRNA can last longer before it's degraded. So RNA processing is really the last thing that occurs in the nucleus, because after RNA processing, this mature mRNA will leave the nucleus. So we can kind of put a line here. 1, 2, and 3 are things that occur in the nucleus. Okay? Now, what about if you've made your mRNA, but you no longer want to make that protein? Okay? So this is something that can occur in the cytoplasm or outside the nucleus. In RNA interference, okay, there are a couple types of tiny RNA. They're called microRNA or siRNA. MicroRNA and siRNA are tiny pieces of RNA. They can do a couple things. First of all, they all bind to mRNA. after they bind to mRNA, two things can happen. Either 2A, they can degrade the mRNA, or 2B, they can block translation. So for instance, if this right here is the mRNA, okay, these could be these Cy or microRNAs that are blocking translation. Or, again, if this is the mRNA, okay, these could be the Cy or microRNAs that are causing enzymes to come along and break the mRNA into tiny pieces and causing it to be degraded. So RNA interference is basically preventing the mRNA from being translated. Okay, so it's basically preventing translation. So if you make an mRNA and you don't want to make its protein, you can prevent translation here. The next example, and again, we're all about things that are occurring in the cytoplasm or in the organelles outside of the nucleus, is ribosomal regulation. In the ribosome, mRNA can be blocked by regulatory proteins. So again, here's our mRNA. Okay, this is supposed to be our ribosome right here. And what can happen if you want to prevent translation is you can have this regulatory protein joining to the mRNA and preventing the large subunit of the ribosome from attaching. So that can prevent translation as well. The next step we've already talked about as well. Okay, so again, this is preventing translation. 6 is after translation. So remember, protein processing happens after translation. This is also called post-translational modifications. Okay, this can happen in the cytoplasm. This can happen on the ER. This can happen on the Golgi. So what can happen to these proteins? They can be phosphorylated. We add a PO4 which activates them and energizes them. This is one thing that can happen to proteins after they've been made. A second thing that can happen to proteins after they're made is glycosylation. This should be a Y actually. Okay, glycosylation. We can attach carbohydrates to proteins, right? So we talked about this before. If this is our protein, we can add this carbohydrate group to it to make a glycoprotein. This makes membrane proteins, like the MHC proteins we talked about in the immune system, self or non-self proteins. Another thing that happens after translation is the protein folds correctly, from primary to secondary to tertiary, and maybe even to quaternary structure. This gives the final structure of the protein. Okay? And there's actually a protein that helps proteins fold. So it's called a chaperonin protein. This helps proteins fold up. Okay? So that happens after the proteins are made. Not all of these things happen to every protein, but these are things that can happen to proteins. Okay? Finally, protein degradation. When proteins get old, they need to be, I hope you guys aren't seasick with me moving my camera around. Protein degradation just means breakdown of old proteins. Okay? Basically what happens first is there's an enzyme, or there's a protein called ubiquitin. So ubiquitin is a protein, and it produces a tag that tags proteins for destruction. Okay? So like if this right here is an old protein, ubiquitin is attached to this old protein, and that tags it. That says this protein needs to be destroyed. And then what happens next is an enzyme called a proteasome, it's a really big structure, destroys the tagged proteins. So here's the proteasome right here. It's kind of like a trash can, and this old protein goes in and is broken down, and amino acids are released. They can be like recycled to make new proteins. So these are all the ways we can regulate protein production, protein processing, and protein degradation in our cells, in eukaryotic cells. I will produce another video on prokaryotic gene regulation. Thanks for listening.